Hi, SEA Symposium. This is Salem State University. I'm Dr. Amy Sprenkle, and our presentation is the discovery and annotation of Microbacterium foliorum phage juicer and Gordonia rubripratincta phage geek. Brahim Azeni, Mary Bilbo, Robin Cashins, Christina Gonzalez, Nick Mignot, Mariah Panza, Sergio Trevino worked on the annotation, and Robin is going to start us off.
Salem State University has so far annotated 6 phages, 2 from the Genome Exchange and 4 phages isolated by students in the program. Microbacterium foliorum was isolated from grass in Germany and has a yellow pigment. It is one of more than 90 species that are in the genus Microbacterium, which are small gram-positive rods, giving them their diminutive name. They are found in dairy products and when in cheese rinds, add to the distinctive flavor of the cheese.
Gordonia is a gram-positive rod-shaped soil bacterium, but it is more acid-fast in nature and more closely related to Mycobacterium species, as shown boxed together in the phylogenetic tree. Gordonia species are attractive for use in bioremediation and biotechnology, and are also known to have caused opportunistic infections in humans. We continue to accumulate data on phage diversity in the phylum Acinetobacteria in hopes of being able to apply this to future understanding of phage biology and for use in treating antibiotic-resistant bacterial infections through phage therapy.
The first phage we discovered was Geek on Gordonia. Gordonia was a new host for us this year, and its acid fastness was demonstrated in much waxier growth morphology and unevenness in the lawns. Once we got the DNA back, we knew that Geek was in cluster CT, was 47,537 base pairs in length, and showed typical organization with the structural genes in the left arm and control genes in the right arm. An exception to this synteny was in cluster CT phages is that the lysine B gene occurs in the right arm rather than in a lysis cassette with lysine A and holin. The typical program translational frame shift occurred in the tail assembly chaperone. The second phage we discovered was juicer. Microbacterium juicer was in cluster EA, subcluster EA6, and was 41,099 base pairs in length. It also displayed the typical genome organization with the structural genes in the left arm and the control genes in the right, with the program translational frameshift in the tail assembly chaperone.
During the fall of 2019, we isolated another Ancylobacterium phage on Microbacterium named Atrexy. Atrexy is in cluster EK2 and was unusual because it was a Protoviridae, which lacks the long flexible tail of the siphon Viridae. Atrexy also demonstrated unusual genome structure. The structural genes are in the right arm rather than the left, with a larger gene of unknown function in the forward direction in the center of the genome. The large gene has no known function. There were no obvious genes indicating a temperate life cycle, but the cluster life cycle is listed as unknown in phages DB. Atrexia had a slow-forming cloudy plaques, and an area of future study is to continue to explore the life cycle of this unusual phage. This concludes the presentation for Salem State University. Thank you for the opportunity to present at the 2021 SEA Symposium.